I'm sure that's going to be very much away from what you guys normally deal with. So the previous talk was about spinal radiosurgery. Of course, I thought that meant spinal cord. And I just wanted to make the point that the spinal cord actually moves. And this is what happens to the spinal cord with cardiac pulsations. So if you do try to do radiosurgery to the spinal cord, or you're trying to map out avoiding the spinal cord, just bear in mind that it actually moves. So with arteriovenous malformations, what they do is they bleed into the brain. And this is a man in his 20s who didn't know he had an AVM and presented with a hemorrhage into the dominant hemisphere. And if he doesn't die, he's going to be left aphasic and paralysed on the right side. So it's a bad disease. You can see them on MRI, the best seen with angiography. So that's the internal carotid artery. This is an AVM in the frontal lobe. And the point about this type of AVM is that that's pretty easy to treat because you can lop off the frontal lobe and nobody notices. Provided it's just one side. And then the patient's cured, okay? So that AVM is not a problem. This AVM is occupying most of the cerebellar hemisphere, and when you inject into the vertebral artery, you don't even get contrast into the basilar. It's just all being sucked into the AVM. And so that's a pretty formidable lesion. But what we can do with that sort of lesion is get the INR doctors to put the catheter right up and block off some of the vessels with glue, left with still a difficult AVM, but now we can take that out surgically. So we can treat AVMs with that kind of thing. I had to borrow this slide. This is an AVM that's in the brainstem, and that's clearly not something that can be operated on, nor could you put a catheter in there and embolise it with glue. So that's a great case for radiosurgery. It's small, certainly amenable to treatment with gamma knife or LINAC radiosurgery. And what we know about radiosurgery for AVMs is that if you deliver a high enough dose, there is a very good chance that the AVM will obliterate. And that's shown in this graph. So if you give about 20 gray marginal dose to an AVM, no matter how big it is, then you will have an 80% chance of obliterating the AVM. It may take 3 years, but that's the likelihood of it going. The problem is if it gets beyond 3 centimetres or 2.5 centimetres in size, you guys will know better than me, the capacity to deliver a 20 gray marginal dose without getting these shoulders of the margin sloping outwards and delivering radiation to the surrounding brain becomes more difficult, right? That's as I understand it. I'm just a simple neurosurgeon. So that's great for this kind of AVM. But it's not great for that kind of AVM. So when you've got an AVM that's this big, it's not for surgery, it's not for embolisation, and it's not for radiosurgery, right? So this is the problem. There are large numbers of people, in our perspective, with this kind of problem where they're currently untreatable. And so if you think about the kind of treatments that are available for AVMs currently, and you think about the pros and cons of treatment, there's about a third of patients or more who we would consider currently untreatable with those current techniques. And so the question is, how can we improve treatment for those patients? Can we improve surgery? No, we're already perfect. And there's no way that we can ever operate on that kind of AVM and the patient survive in a conscious state, right? It's just not going to happen. Endovascular, I mean, I guess if you talk to the endovascular guys, they will say, yes, we'll get better catheters, better glues and stuff. But the reality is that there are hundreds of feeding arteries going into this lesion and many draining veins, and you cannot, in reality, get glue to block off all of those vessels without causing infarcts in the adjacent brain. So that's not really likely. Is radiosurgery going to improve? Well, maybe you could get better physical delivery mechanisms so that you can expand the size that you can treat with that sharp drop-off. That's possible. Alternatively, could you pursue a biological response? And that might be increasing the cell death response to radiation with some sort of radiosensitization, or the opposite, increasing the proliferation of the smooth muscle cells and endothelial cells that we think are the underlying biological response to radiosurgery that occludes vessels. Or could you induce thrombosis? There are some other theoretical kind of concepts. You could use anti-angiogenic factors because it's likely that angiogenesis plays a role in AVM development. But the problem is, once it's got to an AVM like this, it's not an angiogenic problem. It's already happened. And so if you block angiogenesis, it's not going to make it go away. Could you use gene therapy? Is there an underlying genetic problem that's making it do that? Possibly, but again, once it's there, it's not going to go away with gene therapy. So we come back to this idea of stimulating thrombosis, intravascular thrombosis, just within the AVM vessels, as a potential way to induce a biological treatment for AVMs. And this is the concept now of vascular targeting. So if you target, you're trying to get vessels to thrombose in a specific location. So targeted vascular thrombosis. And you can do that if you know that in the endothelial cells lining the vessels that you want to occlude, if there are unique markers, and that's what we're showing here on this diagram, you could potentially target those molecules, usually proteins, with an antibody attached to something that stimulates thrombosis, such as thrombin or tissue factor. So you could theoretically inject something systemically that attaches only in the vessels that are abnormal and induces thrombosis there. And this is not a new concept. I mean, you guys probably know about this. So this is 20 years old. Dennis Hallahan from University of Chicago, showing that you could induce thrombosis in tumour vasculature because the tumour vessels are abnormal. They're not like systemic vessels. You can target them in various ways. It doesn't have to be antibodies. If you know that the endothelial cells have externalised or translocated phosphatidylserine, which is the phospholipid that's normally on the inner leaflet of the cell membrane, right? If it flips to the outside, as it does in apoptosis, but not just in apoptosis, in other cell responses, if it flips to the outside, you then get this situation where you can induce thrombosis through a combination of lipopolysaccharide and actually tissue factor, inducing a thrombotic cascade. So long as you know you've got a situation where the endothelial cells have translocated phosphatidylserine, you can induce thrombosis. So there's ligand-directed and non-ligand-directed. So the question then is, are AVM vessels abnormal? Well, in fact, they are. So the endothelial cells are abnormal. So in a normal vessel, there's a single layer of endothelial cells. But in AVMs, there are multiple layers. The endothelial cells have become multi-layered, they're heaped up, they have what's called endothelial cushions, they express growth factors, particularly on their surface, and so they are abnormal. The problem is that that abnormality just is a graded transition from normal vessels through to AVMs, so that the AVM nidus we want to get rid of is that, but the endothelial abnormality progresses along the feeding arteries to get there. So if you gave a systemic treatment targeting those, you would thrombose those vessels, which would then cause infarction of surrounding brain. So you just want to target that volume of vessels. And we did some work, and others did too, looking at the molecular characteristics of AVM vessels compared to normal brain vessels, so particularly looking at selectins and other adhesion molecules, which are the things that are likely to be abnormal on the endothelial surface. And we showed there are some differences, but they're just not enough to target in a constitutive basis. So could you induce a molecular change in that volume of vessels that you want by using the gamma knife, right? So that's the basis of what we're trying to do. So if we target that volume with radiosurgery, whether it's LINAC or gamma knife, does that change the molecular characteristics of the endothelial cells in that volume such that you can now target them with a ligand-based or non-ligand-based therapy? And you may say, well, that might still limit it to a 3 centimetre volume, but the idea is that if you could induce that molecular change with a lower dose, your physics would allow you to treat a larger volume and not damage the surrounding brain. Okay. So that's the idea behind the research we've been doing. And we can't do it on humans, right? We can't do these experiments on humans. And that's difficult to do in rats because we need to have an AVM in an experimental model because, as I showed you, the endothelial cells in human AVMs are not normal. And if you want to look at the responses of endothelial cells to radiation, you have to actually study the phenotype of interest and its response, because endothelial cells are all different in the different organs, and they respond differently to radiation. I'm sure you know better than me. So we need an animal model, and we need to have cell culture to make this a feasible kind of experimental work. So we developed this rat model of AVM, because basically an AVM is arterialized veins. Okay, it's vessels that are not normally structured to carry arterial high flow, high pressure blood, but have changed accordingly. So we connect the jugular vein to the carotid artery in rats, and here's some angiograms in rats. So this is the carotid artery in the rat, and this is over time showing that what we develop is a nidus with a feeding arterialized vessel. So if I get you to think about this as the nidus, or the collection of vessels that's modelling the AVM, then it's angiographically just like a human AVM. It drains across, so the arterial blood comes up here, comes through the nidus, across the other side, down the jugular vein, across the other side. So we've got this arterialized collection of vessels, and we showed that compared to control vessels, we get the same sort of morphological changes that you see in human AVMs. There's multiplication of endothelial layers, so you get this heaping up of endothelium, you get splitting of the internal elastic lamina, which are all morphological characteristics that you see in human AVM vessels. So morphologically the same. Without going into all the details, we studied the molecular characteristics of interest and showed that the model replicates the human molecular characteristics, the phenotype of the endothelial cells, as closely as possible. And we did some primary cell culture. So these are endothelial cells from that rat model. So that's the AVM phenotype that we've got in primary culture that we can study with radiation. And we can look at the molecular responses using immunocytochemistry in that primary cell culture. So we've got now the basis to be able to study these things. Next thing is we have to deliver radiation, right? So you can radiate cell culture, all right, just put the plate underneath the beam, but for the rats, we need this kind of thing. We did start with LINAC, but we're currently using Gamma Knife. So what we do is, here's the rat with the AVM model. We do a CT scan on every rat and plan it just like you would for a human. Michael Grayson and Back's done a great job helping us to do this 3D reconstruction. So we deliver radiosurgery to the rat AVM. And that means we can now study the molecular changes that occur in the radiated endothelial phenotype of an AVM. Now these, immunohistochemistry obviously doesn't show up very well on slides, but we looked at the adhesion molecules and we showed that E-selectin was increased a bit compared to the control jugular vein. Because E-selectin is not normally constitutively expressed on the surface in brain endothelium, whereas it is systemically, right? VCAM had an increase that was more prolonged. But what was most interesting was that phosphatidylserine is externalized. So that's a control vessel. And what we're seeing here is red is an exon that has been injected systemically before sacrifice. So this is only going to detect phosphatidylserine that's externalized or translocated to the cell surface. Green is von Willebrand factor, so that's the endothelial cells. And what we're showing here is that there is phosphatidylserine externalized on the endothelial cells on the lumen. That doesn't mean they're apoptotic, because they're alive. This is at 3 weeks after radiosurgery. They change in response to radiosurgery, and that's been shown in other organs as well. So that means we could use this non-ligand approach. So we did that, and so just to refresh your memory, if you've got externalized phosphatidylserine and if you inject lipopolysaccharide and tissue factor, that should induce thrombosis in the vessels that have the translocated phosphatidylserine, which is only where you've done the radiosurgery, right? So this is systemically injected stuff. These are control animals, so this is an AVM model. There's no blood in the vessels because we've washed it out, right? This is a treatment animal where we've done the same thing except we've injected the lipopolysaccharide and tissue factor, and we get this widespread thrombosis. And it doesn't occur anywhere else except in the volume that we've radiated. This is just to show that it's a pre-mortem thrombus, not a post-mortem thrombus. And Raj Reddy, some of you may know, did this work for his master's degree and showed that this thrombus was durable. It lasted for as long as we studied them, which was 3 to 6 months. The problem is that, so you might say, why don't we just go and inject humans with lipopolysaccharide? Well, the problem is they'll go into septic shock, right? So you can't really use that in humans. And so we really want to pursue a ligand-directed technique. And although you might say, well, the externalized phosphatidylserine is a good target, it's possible that there are other targets. There might be proteins that get expressed on the surface that are much more discriminating and easier to target than the phosphatidylserine. And so we thought it was important to do a very broad screen for the molecular changes that occur and see if there was anything that could be particularly targeted that would be more specific and more durable. And so for a target to be useful, it has to satisfy 3 criteria. It has to be discriminating. It has to be much more highly expressed on the surface than in the systemic vessels. That's kind of obvious. And ideally, it would be a molecule or a protein that's not expressed systemically at all. So very highly discriminating. It has to be on the surface. We can't have something that just increases in the cell cytoplasm because that's not accessible to the thrombotic technique. And it has to be that it changes only where you've done the volume of radiosurgery, so you're being very precise about where you can create the thrombosis. And so there are techniques that we use to study each of those things. This is our proteomic technique. One of the problems with studying protein changes on the cell surface is that the cell membrane, the proteins in the cell membrane, account for 1% or less of the proteins in the cell. So you can't just take this, and then the endothelium is only a small amount of the vessel wall, right? So you can't just take the vessel and mince it up and look at the proteins, because the proteins on the endothelial surface are a tiny fraction of the total proteins that you would get out of the minced up vessel. So we've used this biotinylation technique. So what we do is we, either in the cell culture, put biotin over the living cells, or in the animal model, inject biotin. The biotin binds to the proteins that are only on the surface, and then you can do proteomics looking at biotinylated proteins. So this is just the cell surface proteins. And if any of you have done proteomics, like you get this enormous printout, right, and you look at how many changes, how many fold changes there are. And so I don't want to go into all that detail, except to say that we come up with a list of proteins of interest, right? Things that are increased on the cell surface, some of which are usually only in mitochondria. They're not normally on the cell surface. So it's a really interesting kind of biology to start with without even thinking about where we're going with it. So some proteins that are normally on the mitochondrial membrane are now on the outside surface of the endothelial cells. And we've shown, I'm not going to go into that now, but we've shown that they really are, using immunohistochemistry techniques, they really are on the cell surface. But so that's the program that we've been undertaking. We do PCR. Now, obviously, this is affected by the total cell protein, but we want some sort of quantitative validation of what we're getting on the proteomics. And interestingly, the E-selectin that initially turned out looked promising, probably on the PCR, is not so discriminating compared to normal vessels, whereas ICAM looks very promising in terms of the change in protein expression, as well as VCAM. So ICAM is a protein that we're pursuing. E-selectin we're not so keen on anymore. We need to know that the protein changes are on the cell surface, so we use immunocytochemistry using confocal microscopy that doesn't give us quantitative information but tells us where in the cell the protein changes are. So we can see which changes here, for example, PCAM, pretty clearly on the cell surface, not just in the cytoplasm. So for each of the proteins of interest, we look at each of these things. A technique that we think is very useful is the in vivo molecular imaging. So having delivered the radiosurgery to the rat model, we then take the live rat, inject it with, in this case, a fluorescent tracer, but it could be a radioactive tracer that you put into a PET scanner. This is optical imaging. So in this case, it's a fluorescent tracer tagged to a nexon. So you inject it into the live rat. It binds to cells where they've got phosphatidylserine externalized, and then you shine a light to them externally and you get the fluorescent signal back. So that's a live animal showing how much change there has been of the molecule of interest, in this case phosphatidylserine, and we can get this three-dimensional plot. So we get, for the first time, a quantitative analysis in a volume sense of the specific molecular change from radiosurgery. And that shows that it's really just where we radiated. It's nowhere else, right? And it gives us an idea of the quantification as well. And we can put that into a graph. So this is showing, in fact, what was interesting in this research was that the non-radiated AVM vessels did actually translocate phosphatidylserine as well, but they did it more once they were radiated. So there is a statistical difference between the radiated and non-radiated vessels, particularly at the 3-week mark and beyond. So whereas we were initially thinking that we might target our treatment in this early phase, for the phosphatidylserine, we're thinking it should be up 3 weeks plus. The next step is to identify or work out the best way to deliver a prothrombotic ligand, and we don't want to put thousands of rats through this kind of experimental work, so we have an in vivo setup. So we have this setup where there are endothelial cells in culture. We have a blood circuit being pumped around with a blood pump, so it's mimicking the circulation. We radiate the cells and then inject into that circuit the conjugate of antibody and prothrombotic molecule. And the example I'm going to show you is where not an antibody, but we've used an exon because that binds to phosphatidylserine, conjugated to thrombin. Right? So we can then study what happens to the endothelial cells and whether platelets stick to the endothelial cells. So what this shows, if I just show you these two, this is a control cell culture. So these are endothelial cells. Platelets are tagged red. So if we don't radiate the endothelial cells and we inject in culture and we inject an exon tagged to thrombin, platelets don't stick to the cells. Whereas if we radiate them, that conjugate sticks to the platelets stick to the cells. Okay, so it gives us a method of quantifying how good a conjugate is at sticking to the cells and producing platelet adhesion and hence thrombosis. So we can screen through various prothrombotic conjugates and choose the best ones to take through to the animal model. And what we're working on at the moment, which I can't show you any data for just yet, but is using nanoparticle technology. So instead of having a single molecule conjugate, we'll have the antibody connected to a carbon nanocage with multiple thrombin or tissue factor molecules. So we'll amplify the thrombotic response. And this is going to allow us to really test that quite quickly. This is the picture I showed you before, which was the non-ligand approach. What we're currently doing now, and these are a couple of neurosurgery trainees doing some research, is this is a rat, and there's the catheter coming up here and injecting in contrast. What I'm going to show you next is hot off the press. So this is an experiment. We're underway looking at groups of animals using the annexin conjugated to thrombin as the prothrombotic treatment. In these pictures, the images are flipped. So this is actually the animal's left-hand side. There's the transfemoral, we've got a femoral angiogram in the rats. So the catheter comes up here. We're about to inject contrast. The nidus is here. It's actually on the animal's left. So what you should be able to see, is the contrast filling the nidus and coming down the other side. Right? So that's a nidus that has been radiated with 20 gray, marginal dose, and it's a little bit fuzzy, I know, but on our screen it's perhaps easy to see. The nidus is still there. The blood still flows through the other side and drains down the draining vein. This is a treated animal, so this is the same experimental setup, but the only difference is that the animal has been injected with annexin conjugated with thrombin. And what we see here is that, okay, so as the contrast is injected, a little bit goes into that feeding artery, but most of it comes back down the subclavian, and there's none that goes down the other side. I haven't yet got the histology to show you, but that's fairly good evidence that there's thrombosis within the nidus, and that would fit with our previous experiments. And it shows that phosphatidylserine is still probably quite a promising target, and the annexin approach rather than the lipopolysaccharide is a much safer kind of technique. So that's where we're up to, and I guess I'd like to close by just saying, I think we think this is a pretty promising kind of field. We've got some encouraging targets that we're processing through. I think the nanoparticles are going to be really important, so we're developing those treatments. But it doesn't just have to be AVMs. It could be anything anywhere in the body where you could use radiosurgery to define a volume within which you now have molecular changes on the vessels and you could deliver anything. It doesn't have to be prothrombotic. It could be a chemotherapeutic agent, something that gets stuck on the vessel to the molecule of interest and is now delivered locally. This is obviously a big team effort over a long period of time. I was reminded, having a beer with Michael, that Michael Grace is not on this list, so you have to give me a photo to put on the list. And we've obviously had a bit of funding, including from Electa, to help us. Electa gave us funding to transition from the Linac to the Gamma Knife, which was very helpful to get that program moved over. But most importantly, it's lots of people who get involved, get very passionate about it, and I'd like to just pay tribute to the work that they do. Andrew, I've got you on there, haven't I, somewhere? I'm sorry if I haven't. I'll have to get your picture as well. I try to keep up, but there's a lot of people. So if I've missed anyone, I'm sorry, but thanks very much. That wouldn't get past ethics. No. So where I think, once we get through the rats and probably a large animal model, like let's say ICAM turns out to be the best target, and we've got some sort of nanoparticle technology that can promote thrombosis. We've got something, and we want to take it to clinical trials. Well, the first thing would be to show that ICAM, or whatever it is actually expressed in a human AVM when you give radiosurgery. Well, the way we could do that is using PET imaging on patients who actually have radiosurgery. Now, I think that would get through ethics. I don't think the radiosurgery prior to surgery would get through. And we've looked at circulating microparticles as a means of getting a window to what the endothelial changes are. Andrew's been involved in that. It's technically pretty difficult, and I'm not sure it will really give us very good quantitative data. I don't think anyone's doing anything apart from this. But that goes to the radiosensitization idea of increasing the innate. I think what we're proposing here is quite different to what actually happens when you treat someone with radiosurgery. It's not really speeding it up. It's adding something different. What you're suggesting is more along the lines of speeding it up. And it doesn't have to be, like, you can get nanoparticles that release their contents if they're light activated. Now, getting the step from there to being radiation activated and releasing contents is a pretty small step. That we've looked at that because we talked with our physicist and nanoparticle technology people, and they said, you could just use those nanoparticles and then put a laser beam up through the AVM. I'm going, no, that's not going to work. So we're sticking with the idea of a nanoparticle that just increases the ratio of effector molecules to the ligand.
Super interesting. That was fantastic. So thank you very much.